Hello everyone, I am Swati from BSc Chemistry on our final year. Today my chapter is Bioinorganic Chemistry. Inorganic chemistry deals primarily with salts and complex metal ions, whereas bioinorganic chemistry deals majorly with inorganic chemistry when applied to biological systems. Thus, bioinorganic chemistry can be regarded as the branch of science that deals that relates inorganic chemistry with life processes by recognizing the role of metal ion in the normal growth and function of biological processes. In brief, bioinorganic chemistry highlights the role of metal ion in biology. Bioinorganic chemistry is a rapidly developing area of chemistry because of its importance in biological system. The essential biological role of a few elements have been known for many years. This chapter will focus on the important role of metals, non-metals, and their compound in biological processes. Essential and trace elements. We think that the chemistry of life is solely composed of organic molecules. It is a wrong concept. In fact, there is a long list of inorganic compounds that are essential to living systems. For example, the skeletal and integumental materials outer covering in higher animals is composed majorly of inorganic materials, and the lifetime consumption of oxygen by a living entity is a major fraction of its material needs. In addition, water, oxygen, calcium phosphate, and calcium carbonate are the major inorganic constituent of living matter in terms of quantity. Table 5.1 List the abundance of element in earth crust and in ocean compared with some abundances in typical mammals. This is the table. Of all the known elements, it is about 30 elements that have been recognized as essential and indispensable to life. Out of the 30 elements, 15 of them are metal. The essential material are needed for the growth and normal function of animals and plants. As absence of these elements result in severe malfunctioning of the living organisms. Thus, an element may be considered essential when its insufficient intake produces an impairment of normal functions, and its restoration relieves the impaired function. Some of these elements are required in bulk or macroscopic amount to all forms of life and are classified as bulk elements. Some of the elements are required in bulk or macroscopic amount to fill forms of life and are classified as bulk elements. Some of the bulk elements are required for structural purposes, skin, bones, egg cell, various organs, while the rest are used as minerals. In other words, in the absence of essential elements, the organisms can neither grow nor complete its full life cycle. The essential elements can be further subdivided into two groups: bulk elements or trace elements. Bulk elements, essential elements that are formed in higher concentration. Trace elements, essential elements that are present in low concentrations. Bulk quantity of group 1 and 2 metal are required mainly to balance the electrical charges with the negatively charged macromolecules in the cell and also to maintain the osmotic pressure in the cell. The reaction flask of a living organism is the cell. In addition, the transition elements is the heavier member of the P group, also play an important role in the bioinorganic chemistry. Some of the inorganic elements are required in small quantity and are classified as inorganic trace elements. These different types of essential elements present in living system are classified as table 5.2. It is important to note that all the elements displayed in the table are required by all its plants and animals. For example, sodium ion Na positive is of special importance to higher animals. It is heavier, essential to many bacteria and plants except blue-green algae. The ultra trace elements VIZ, aluminium and vanadium are needed by some higher plants. Molybdenum is essential to only those animals which derive their nitrogen from inorganic sources. Selenium is an essential micronutrient for many species but prove to be toxic at higher levels. Selenium deficiency in human result in a dependent condition of heart tissues known as Kishan disease, named after a country in China in which endemic because of extremely low selenium level in the soil coupled with a population that entirely upon locally grown grains. It is important to note that even some essential elements may prove to be toxic if consumed higher quantity. Essential bulk elements: sodium and potassium. We have learned that both sodium and potassium are essential for life processes. For example, at least 1 gram of sodium ion per day in our diet. So, however, because of our habits to adding salt and some people consume as many as 5 times more than required. However, intake of potassium create problem. Instead, potassium deficiency is more common. We must include potassium rich foods like bananas and coffees in our diet. In inorganic chemistry, we always think of the similarities between sodium and potassium. However, in biology, it is the difference between sodium and potassium which plays important role. Cells pump out sodium ions of the cytoplasm, whereas the cell pump the potassium ions inside. This ion transport is sometimes called a sodium pump, and it involves both the active expulsion of Na positive ion and the active take-up of K positive. It is the difference in the total alkali metal ion concentration inside and outside the cell that produces an electrical potential across the cell membrane, which is important for the functioning of nerve and muscle cells. Concentration of ions: red blood cell Na positive 11, K positive 92. Blood plasma Na positive 160, K positive 10. The transport of ions required energy, and this is obtained by the hydrolysis of ATP, adenosine triphosphate. It is estimated that hydrolysis of one ATP molecule to ADP provides enough energy to move 3 Na positive ions out of the cell and 2 K positive and 1 H positive ions back in the cell. Calcium and magnesium. Both calcium and magnesium are present in body fluids. Mg2 positive ions are concentrated in the animal cell and Ca2 positive ions are concentrated outside the cell. This is similar to the presence of K positive ions inside the cell, whereas Na positive ion is outside the cell. Calcium 2 positive ions are present in the external skeleton of creatures such as shellfish and corals. The material used is calcium carbonate. Creatures with interior skeletal such as mammals and reptiles use calcium hydroxide phosphate Ca5OH Pa4 whole thrice, and the animal on teeth as CaF2. Both these compounds are highly insoluble. Important minerals used in biological materials are listed in table 5.4. Ca2 positive ions are important in blood clotting and are required to trigger the contraction of muscles such as those that control the beating of the heart. In fact, some type of muscle cramps can be prevented by increasing the intake of calcium ions. A decrease in calcium 2 positive content of blood, either due to dietary deficiency or from an insufficiency of the parathyroid hormone, may lead to a disease called tetany. On the other hand, an increase in the concentration of Ca2 positive ion in the blood may lead to calcification of several internal organisms. The absorption of dietary Ca2 positive and or Mg2 positive ions from the intestinal tract may be prevented by the simultaneous ingestion of organic acid, example oxalic acid, with which it combines to form an insoluble salt. It means that the availability of dietary Ca2 positive is greatly affected by the other constituent of the diet. Both Ca2 positive and Mg2 positive ions are excreted mainly via the large intestine instead of the kidney. Magnesium, because of its high charge radius ratio and consequent strong hydration. As in magnesium, H2O6 2 positive plays important biological roles that are different from that of calcium. It acts as a counterion to the negatively charged group in nucleotides and polynucleotides. One of the most important nucleotides is ATP, adenosine triphosphate, which serve as the fundamental energy source for the living cells. ATP is thus known as energy-rich molecule and is used to derive most of the chemical reaction of the cell. Mg2 positive ion form a complex with ATP. It has been observed that all transphosphorylation reactions involving ATP proceeds smoothly in the presence of Mg2 positive. ATP is an anhydride of the phosphoric acid. The hydrolysis of anhydride is a very favorable reaction. Thus, the hydrolysis of ATP to ADP shown in the equation below liberates 30.7 kJ per mole, 7.3 kcal per mole of energy at pH 7. The living system thus utilizes this energy to drive energy requiring biochemical processes. It has been observed experimentally that the activating effect of Mg2 positive and transphosphorylation may be duplicated in vitro by Mn second, Ca2 positive, CO2 positive, Zn2 positive, or Ni2 positive. The most important aspect of the biochemistry of magnesium is its role in photosynthesis. Magnesium containing chlorophyll, using energy from the sun, converts carbon dioxide and water into sugar and oxygen. Without the oxygen from the chlorophyll reaction, this planet would still be surrounded by a dense layer of carbon dioxide. In addition, without the sugar energy source, it would have been difficult for life to progress from plants to animals. It is important to note that magnesium ion sits in the middle of the chlorophyll molecule and holds the molecule in a specific configuration. Magnesium has only one possible oxidation number +2. As a result, the electron transfer reaction involved in photosynthesis can proceed without interference from the metal ion. Phosphate. Phosphorus is an important element for life processes. For example, free hydrogen phosphate PO4 3 negative and dihydrogen phosphate H2PO4 ions are involved in the blood buffering system. Phosphate is the linking unit in the sugar ester of DNA, deoxyribonucleic acid, and RNA ribonucleic acid. The phosphate units make up part of ATP, the essential energy storage unit in living organisms. Bone is a phosphate mineral. The commonly called mineral is calcium hydroxide phosphate. The presence of phosphorus in bones and its function in regulation of pH of blood constitute an important feature of phosphate. It is important to mention that the normal formation of bones depend upon the relative amount of dietary phosphorus and calcium in addition to vitamin D. Essential trace elements. Among the known trace elements, the three most prominent biologically active metals are iron, copper, and zinc. The remaining trace elements, excepting boron and fluorine, are used in a very small amount, less than 10 mg in adult human, and are referred as ultra trace elements. The trace elements are known to play an important role as metalloenzymes. Iron is essential in small amount for both plant and animal life. However, like copper and selenium, it is toxic when used in larger quantity. Biological iron is the most important transition element and is found in greatest abundance in biological system in the form of porphyrin complexes or HAMs such as hemoglobin and myoglobin. Porphyrins are macrocyclic ligands and the derivatives of porphyrin, discussed later. A human body weighing about 70 kg contains about 4.3 gram of iron. Nearly 70% of this iron is present in hemoglobin and about 3% in myoglobin. The remaining 23% of body iron is present in ferritin. Ferritin is present in spleen, liver, and bone marrow. It consists of a cell of linked amino acid peptides called apoferritin surrounding a core of an iron third oxohydroxophosphate. The core is a cluster of iron third ions, oxide ion, hydroxide ion, and phosphate ions with the composition FeOOH8, FeOPO3H2. It is very large, containing up to 45,000 iron ions. With its hydrophilic coating, this large aggregate is water soluble and is concentrated in the spleen, liver, and bone marrow. Apoferritin protein can be prepared from ferritin by reduction of ferric ions to ferrous ion at pH equal 4.5, followed by the removal of Fe2 positive by dialysis. The formula weight of apoferritin is nearly 46,500. Ferritin can also be synthesized in the lab with properties similar to natural ferritin. It can be prepared by treating crystalline apoferritin with an inorganic Fe2 salt in the presence of oxygen. Ferritin is probably formed in vivo by an analogous process. The various steps involved in the pathway of Fe2 positive from the foodstuff to hemoglobin and to ferritin involve the following processes. The Fe2 positive of the bond dietary material is reduced to Fe2 positive in the gastrointestinal tract. After absorption into the cell of intestinal mucosa, Fe2 positive is incorporated into ferritin as Fe3 positive. The Fe2 positive in mucosa is also converted to Fe3 positive plasma, bounded by iron binding globulin named as transferrin. Plasma Fe3 positive is in equilibrium with iron in the liver, spleen, and bone marrow. The following changes that occur are summarized in the SAM 5.1. Zinc. Among the trace essential elements, zinc is secondly only to iron in importance. Zinc is essential to all organisms and play an important biological role in the enzyme system of animals and plants. Humans contain about 2 grams of zinc and is one of the most important and crucial elements in our body. Deficiency of zinc are not life-threatening but contribute to fatigue, lethargy, and related symptoms. Zinc 2 positive ion is a soft acceptor compared to other divalent ion. Example: Mg2 positive, Ca2 positive, or Mn2 positive, and others. Stronger Lewis acid towards many biomolecules compared to Zn2 positive ion. Cu2 positive ion is also soft but additionally intermediary oxidation state. Example: Cu2 positive. Different oxidation state of copper may allow different redox reactions and completed behavior. The complexes of zinc ion Zn2 positive are much more labile than the corresponding complexes of Mg2 positive, Ni2 positive, or Mn2 positive. This facilitates the role of zinc ion in enzymes. Zn2 positive ion is completely resistant to redox changes at biological potential. Its role cannot be affected by changing the redox potential in the organism. A number of enzymes containing zinc ion are known. The most important of all the enzymes are carbonic anhydrase. It is present in red blood cell and is involved in the respiration. It speeds up the absorption of carbon dioxide by red blood cell in muscle and other tissues. On the other hand, in reverse reaction, it involves the release of CO2 in the lungs. The reaction is pH dependent. Thus, the zinc-containing enzyme carbonic anhydrase catalyzes the otherwise slow carbonic acid carbon dioxide equilibrium reaction. The carbonic anhydrase has a common 4-coordinated environment around Zn2 positive ion, in which 3 of the ligands are imidazole, nitrogen of 3-histidine, and the 4th is water molecule or hydroxide ion, as shown below. A water molecule coordinated to a zinc ion has a pK near 7, and thus the role of zinc is to be reduced in the basicity of hydroxide ion such that a high effective concentration of hydroxide can exist in an approximate neutral medium. Another important enzyme containing Zn2 positive ion is carboxypeptidase. It is present in the pancreatic juice and is primarily involved in the digestion of protein by animal and protein metabolism in plant and animal. This enzyme catalyzes the hydrolysis of the terminal peptide amide link at the carboxyl of the peptide chain. The enzyme is selective and works only when the R group in the terminal amino acid is aromatic or a branched aliphatic chain and has the L configuration. Copper. Copper is the third most biologically important transition metal after iron and zinc. Human beings need around 8 mg of copper daily. A deficiency of copper than the optimum value result in the inability of animals to use iron stored in the liver. As a result, the animals suffer from anemia. The copper is bound to protein in the body either as metalloproteins or as enzymes. Excess of copper is highly poisonous, particularly to fish. This is the reason it is always advised not to throw copper coins in fish pools for good luck. Human beings can excrete an excess of copper, but a biochemical defect can result in copper accumulation in the liver, kidneys, and brain. The illness caused by the excess of copper is called Wilson disease. It is a hereditary shortage of ceruloplasma, causing copper accumulation in the liver, kidney, and brain. Patients suffering from Wilson disease are cured by administering ethylene diaminic acid, acetic acid (EDTA), which forms soluble copper complex. The soluble copper complex can be excreted harmlessly through urine. At the same time, many other essential metals involved in other enzymatic reactions from EDTA complexes and are excreted. Thus, the EDTA treatment can upset many unwanted enzyme reactions. As a result, utmost care should be taken in monitoring Wilson disease. Hemocyanin is a copper-containing protein which is important as an oxygen carrier in some vertebrate animals. Despite its name, it is non-heme protein. It is found in the blood of snails, crabs, lobsters, octopus, and scorpions. The oxygenated hemocyanins are blue-colored, unlike human blood, and have one dioxygen molecule attached to two copper atoms. Deoxygenated hemocyanin contains Cu1 and is also blue. Selenium. Selenium is essential to health. All classes of organisms metabolize selenium. In human and other mammals, serious disease arises from either excessive or insufficient dietary selenium. The toxic effects of selenium have long been known, particularly for grazing animals. In soils with high selenium content, some plants accumulate large amounts of selenium as methylselenocysteine in order to keep selenium away from their metabolic machinery. Animals that graze these selenium accumulating plants develop severe toxic reactions, including blind stagers and alkali disease. Selenium poisoning in humans is rare, but has occurred when mines have caused selenium to be leached from the soil into drinking water supplies. Loss of hair and nails is seen in cause of human selenium toxicity, followed by anemia and jaundice. Although selenium is toxic at high level, selenium is an essential micronutrient for mammal species. Selenium deficiency in humans results in degenerative condition of the heart tissue known as Caisson disease. This disease is named after a country in China. The accepted minimum daily requirement of selenium for adult human is 70 micrograms. Many types of food provide selenium, particularly seafood, meats, grains, and the onion family. Mammals and birds require selenium for the production of the enzyme glutathione peroxidase, hydrogen peroxidase, and organic peroxide, which cause oxidative damage to cellular component, are enzymatically destroyed by these selenoprotein. Such antioxidant activity suggests that peroxide and therefore selenium protect against oxidation-induced cancer. Chromium and molybdenum. Trace amount of chromium are necessary in the diet of mammals. Chromium third and insulin are both involved in maintaining the correct level of glucose in the blood. A deficiency of chromium or an inability to use the chromium 3 can result in the removal of glucose from the blood as fast as normally and leads to diabetes. Compound containing chromium, example dichromate or chromate, when ingested or absorbed through skin are carcinogens. Therefore, at most case must be taken while using K2CR207 or K2CR04, especially while performing titrations. Molybdenum is the most biological important member of the chromium family. It is the heaviest, higher atomic number elements to have a wide range of function in living organisms. The most important molybdenum enzyme, which contain iron as well, is nitrogenase. Thus, nitrogenase contain two protein, example molybdenum-ferredoxin and azoferredoxin. It is air sensitive and brown colored species containing two amino atoms, 24 to 36 Fe atom, 24 to 36 S atom, together with a protein. It has a molecular weight of about 2,25,000. Azoferredoxin is yellow, air sensitive, is a derivative of ferredoxin, Fe4S4SR4. It has a molecular weight in the range of about 50,000 to 70,000. The enzyme nitrogenase occurs in bacteria and reduces the inert dinitrogen of the atmosphere to the ammonium ion. This reaction is used in the synthesis of protein by the plants. Let us have an idea of the importance of this enzyme by the typical example. The amount of dinitrogen fixed biologically is estimated to be 175 million tons as per year, compared with a total of 110 million tons of NH3 produced by the Haber-Bosch process and the distillation of coal. Question 1. Why is a metal as rare as molybdenum so biologically important? Answer. There are several reasons to address this question. First, the molybdenum MoO42 negative has a high aqueous solubility at near neutral pH value, making it easily transportable by biological fluids. Second, the ion has a negative charge, making it more suitable for different environments that are the case of cations of period 4 transition metals. Third, the element has a wide range of oxidation state +4, +5, +6, whose redox potential overlaps with those of biological system. Fourth, being a soft Clavis acid, molybdenum shows a strong bonding preference for the soft Clavis base sulfide, another important component of these enzymes. Nickel. The biochemistry of nickel is poorly understood of all those of period 4 transition elements. It is present in some enzymes system in the form of porphyrin type complexes. Some of the tropical trees concentrated nickel to such an extent that it makes up 15% of their dry mass. It is highly toxic to most plants and moderately toxic to mammals. Cadmium. Cadmium is a toxic element that is present in our foodstuff and is normally ingested at levels that are close to the maximum safe level. Kidney is the organ most susceptible to cadmium. Cadmium ingested 100 ppm cause several damage. Cigarette smoker absorb significant level of cadmium from tobacco smoke. Exposure to cadmium from industrial source is a major concern. Use of nickel cadmium battery is becoming a major waste disposal problem. Many reputed companies now accept return of used nickel cadmium batteries so that cadmium metal can be safely recycled. Mercury. Mercury is hazardous because of its relatively high vapor pressure. The mercury vapor is absorbed in the lungs, dissolved in the blood, and is then carried to the brain. This causes irreversible damage to the central nervous system. It is slightly soluble in water because of very weak metallic bonding. Inorganic compounds of mercury are less toxic as they have less solubility in water. Arsenic. Arsenic is an element essential to life. However, we need ultra trace amount of this element. Anything more than a teeny amount causes arsenic poisoning. Arsenic in measurable quantity causes death. through enzyme inhibition. Non-metals. The non-metals among the essential elements are hydrogen, carbon, oxygen, nitrogen, phosphorus, sulfur, chlorine, and iodine. Majority of these are needed in trace amount except C, H, N, and O, which are abundantly available and constituent most of the chemical in the living system. Phosphorus. Phosphorus is an essential amount for life. It constituents an important part of energy-rich molecule known as ATP, adenosine triphosphate. It is also an important component of bone as apatite. Calcium hydroxide phosphate. Partial formation of F strengthens the structure of the teeth and make it less soluble in the acid formed from fermenting organic material in the teeth. Iodine. Nearly 75% of the iodine present in the human body is located at one place. Example, the thyroid gland. It is used in the synthesis of hormones thyroxine and triiodothyronine. These hormones are essential for growth, for the regulation of neuromuscular functioning, and for the maintenance of male and female reproductive functions. One common cause of goitre is a lack of iodide ion in the diet. A symptom of goitre is a swollen lower part of the neck. The enlargement is the attempt of a thyroid gland to maximize absorption of iodine in a iodine deficient circumstances. In order to remove iodine deficiency, potassium iodide is added in the common household salt. Such a salt is known as iodide salt. Chlorine, Cl negative. The chloride ion has a vital role in the ion balance in our bodies. It does not appear to play an active role, but simply act to balance the positive ion of Na positive and K positive. It is a major anion found inside and outside the cell. It is essential as NaCl electrolyte for higher mammals and plants. Chloride ion is essential in digestive juices. Deficiency of chloride ion in infants leads to impaired growth. Nearly 30 elements are now recognized as essential elements for at least some form of life. It includes most of the elements through atomic number 35, except for Be, beryllium, aluminium, scandium, titanium, along with noble gases. Molybdenum and iodine are also essential. It is interesting to note that all elements appreciably abundant in the human body, with the exception of phosphorus, are also abundant in seawater, suggesting without that our family tree has root in the sea. Metalloporphyrins. Porphyrins constitute an important class of bio-inorganic compound in which a metal ion surrounded by 4 nitrogen of a porphyrin ring. Porphyrins are macrocyclic tetradenate ligands made up of 4 pyrrole rings linked together through methane bridge as shown in figure. This tetradentate has a conjugated double bonding system with 4th nitrogen donor site. 2 of the 4th nitrogen are nitrogen, while the remaining 2 nitrogen are secondary nitrogen. The tertiary nitrogen atom is coordinate bond by donating a pair of electrons each to the metal ion by a coordinate bond as shown by narrow head. The other 2 secondary nitrogen lose a proton each and form a covalent bond with metal ion as shown. Thus a porphyrin ring serve as a tetradentate di-negative ligands. As a result, the cations like Fe2 positive or Ni2 positive or Mg2 positive will form a neutral complex with porphyrin, which in the process loses their protons as shown in the figure. When the 4 pyrrole rings of a porphyrin are substituted by organic group other than hydrogen atom called a porphyrin, thus porphyrin are macrocyclic tetrapyrrole system with conjugated double bond. Together with various groups other than hydrogen substituted at the periphery of the ring. The substituent attached to the ring are usually organic group with electron withdrawing or electron releasing characteristic. The complexes formed by the porphyrin with the metal ions, generally D positive, are called Porphyrins. These are biologically important compounds and play a very important role in the animal and plant kingdom. Porphyrin ring system is fairly rigid, most because of the delocalization of electron in the molecule, although the skeleton structure is fairly rigid. Significant variation have been observed among the metalloporphyrins in the interatomic distance between the chiral nitrogen and the metal atom at the center, determining the size of the central hole. From a knowledge of this covalent bond radii, one can estimate that a bond between a nitrogen atom and an atom of the first transition series should be about 2.0 angstrom long. In brief, an ideal size of the atom to fit in the hole is 2.0 angstrom. Thus, the size of the hole in the center of porphyrin ring is ideal for accommodating metal ion of first transition series or lighter alkali earth metal. A metal ion of appropriate size sitting at the center of the porphyrin ring will be surrounded by 4 nitrogen of the pyrrole and forming a square planar geometry. It means that the metal ion and the 4 nitrogen lie in the same plane as shown in figure. However, when the size of the metal ion is smaller than the ideal, the ring will be reluctant to accommodate precisely the metal ions. In this way, the ring will earlier readjust or dissolve itself in order to accommodate the metal ion. On the other hand, if the size of the metal ion is larger than the ideal, the transition metal ion cannot fit in the hole and stay out of the plane of the 4th nitrogen atom. It means that the metal atom sits above the plane of 4th nitrogen atom and acquire a square pyramidal geometry. The nature of metalloporphyrins vary with the nature of the metal ion and the type of organic group attached at the 8th position of the 4 pyrrole ring around the periphery of porphyrins. A group of related compounds, the phthalocyanins shown in the figure, are isoelectronics with the porphyrins. These compounds are of interest because of their intensely colored metal complexes, which are at most commercially application in dyes and pigments. The size of the hole in these phthalocyanins ring is about 0.1 angstrom, smaller than in porphyrins as a result of nitrogen atom compared with carbon. Compare the covalent radii of carbon 0.77 angstrom and nitrogen 0.75 angstrom. Two of the most important metal complexes of porphyrin are chlorophyll and heme, which are being discussed. Chlorophyll is a magnesium complex of porphyrin which play an important role in photosynthesis. Photosynthesis is a remarkable redox reaction in nature that converts water and carbon dioxide into sugar and oxygen in the presence of sunlight. This process is the primary source of food that we eat and the molecular oxygen that we breathe. The function of chlorophyll molecule is to absorb photons in the red part of the visible spectrum and pass this energy of excitation to other species in the reaction chain. The ability to absorb the light is due to the conjugated polyene structure of the porphyrin ring system. The role of magnesium is to make this entire molecule rigid so that the energy absorbed is not easily lost. There are a number of similar but not identical chlorophyll molecules. It is important to note that a chlorophyll, the basic porphyrin system, has been modified in two ways. In pyrrole, the ring IV, one of the double bonds have been transhydrogenated to form a magnesium dihydride porphyrin complex. In addition, a cyclopentaneous ring have been fused to the side of the pyrrole ring third, as shown in figure. In brief, absorption of sunlight by chlorophyll is the first step in the conversion of sunlight into usable energy by plants. It will not to be out of place to say that chlorophylls are nature's solar energy collector. Heme is an iron second porphyrin which is present in hemoglobin and myoglobin, which is being discussed. Hemoglobin and myoglobin. Molecular oxygen play an important role in biological system because of its importance in respiration. The formation of oxygen we have already learnt in nature involve a number of reactions such as redox reaction, electron transfer, atom transfer and Photochemical reaction. A number of proteins are known that bind and transport oxygen to different extents. Such proteins that bind oxygen are called oxygen carrier. The most important and well documented proteins that bind and transport oxygen are hemoglobin and myoglobin. Hemoglobin and myoglobin are very closely related to each other. Hemoglobin is a larger protein with a molecular mass of 64,500 and consists of 4 subunits. Each subunit is made up of a heme group associated with a protein called the globin. It is important to recall that each heme group is associated of an Fe2 positive ion surrounded by 4 nitrogen of a porphyrin ligands. Hemoglobin is a large intracellular protein that is responsible for the red color of the blood cell. On the other hand, myoglobin is a relatively small protein with a molecular mass of 17,000 and consists of one heme group and a polypeptide chain. It is important to recall that the peptide chain consists of 150 to 160 amino acid residue folded about a single heme group. Myoglobin MB is a smaller intracellular protein in vertebrate muscles. Myoglobin was the first protein whose X-ray structure was established in 1959. In 1962, the Nobel Prize in Chemistry was awarded to Candrew and Paretos for their work on the structure of globular protein, including myoglobins. The basic function of hemoglobin and globin are the following. Hemoglobin picks up oxygen in the lungs and transports it to the rest of the body. Myoglobin accepts oxygen from the hemoglobin in the muscles and stores it until needed for energetic process. Deoxygenated hemoglobin uses some of its amino acid to pick up CO2 and then transports CO2 back to the lungs. Oxygen carrier performs the function of acquiring O2 from the atmosphere, transporting it through the circulatory system of the organism, and delivering it to the location where it is used. They all bind O2 rapidly and reversibly according to the equation protein plus O2. This will form protein O2 in the equilibrium. The equilibrium must be largely to the right at the oxygen concentration available at the pickup location, for example, lung air interface, muscle cell blood interface, and mother fetus interface at the placenta. It is important to note that the oxygen carrier interacts strongly with the oxygen to bind it. However, the carrier must not undergo irreversible oxidation in the process. In brief, hemoglobin picks up oxygen in the lungs and transports it to the site of use inside the muscle cell. This oxygen is transferred to the myoglobin, which store it until required for metabolic action, in which O2 is reduced to CO2. Deoxygenated hemoglobin then transports CO2 back to the lungs. Structure of hemoglobin and myoglobin. We have already mentioned that the structure of hemoglobin and myoglobin are very closely related to each other. Hemoglobin is a larger protein and consists of 4 subunits. Each subunit consists of one unit associated with a protein, globin, as shown in the figure. The iron is held in position by coordination with the nitrogen of heme and N complex. This complex reversibly with oxygen as shown in the figure. Thus, hemoglobin is the carrier of blood and the red color of blood is due to the oxygenated hemoglobin. It is important to note that the 4 side units are similar but not identical. 2 subunits is alpha chain of 141 amino acid, while the other 2 subunits form beta chains of 146 amino acid. Chains are coiled together to give a 3-dimensional network. Asymmetric structure of hemoglobin to the relationship among the 4 subunits of the tetramer is shown in the figure. Structure of myoglobin. Myoglobin is a small protein compared to hemoglobin. It consists of a single heme group embedded in a polypeptide chain called globin. The peptide chain consists of 150 to 160 amino acid folded about the single heme group as shown in the figure. The heterocyclic ring consists of a porphyrin containing pyrrole group labeled as A, B, C, D, linked by methane bases. The iron atom in the oxidation state Fe2 present at the center of the heme is bonded by 4 nitrogen of the porphyrin system together with 1 nitrogen atom of the imidazole side chain of the histidine residue, which is a part of the long protein chain of amino acid residue. It is the polypeptide chain that play an important role in the biological fixation of molecular dioxygen. A simple outline of the structure of myoglobin showing heme group embedded in the polypeptide chain is shown in the figure. It is important to note that both hemoglobin and myoglobin have 4 coordinated iron atoms. The oxidation state of iron in Hb and Mb is +2. Iron atom is bonded to 4th nitrogen atom from the chiral rings or porphyrin and the 5th nitrogen from the protein chain. In both the compound, the 6th position is occupied is weakly bonded to water. Hemoglobin and myoglobin is in such molecule are named as deoxyhemoglobin and deoxymyoglobin respectively. However, when molecular oxygen occupies the 6th position, which is trans to histidine chain, then the molecule are named oxyhemoglobin and oxymyoglobin respectively. A myoglobin polypeptide is comprised of 8 separate right-handed alpha helices designated from A to H that are connected by short non-helical regions. Role of hemoglobin and myoglobin. Hemoglobin and myoglobin play a crucial role in transporting dioxygen O2 from lungs to tissues and CO2 as HCO3 from tissues to the lungs. The function of hemoglobin is to bind O2 at the high oxygen partial pressure found in the lung tissues to form HbO2. Hemoglobin carries this oxygen without loss through the blood and that releases it to myoglobin. In other words, the dissociated oxygen from hemoglobin diffuses to the tissue where myoglobin picks it up and stores until it is needed. It means that myoglobin has greater oxygen affinity than hemoglobin at low pressure. The hemoglobin and carbon dioxide are then returned to the lungs where CO2 is exhaled. The sequence of steps can be represented as shown in the figure. It is clear from the same that both hemoglobin and myoglobin play a cyclic role in transporting O2 from the lungs to the tissue and CO2 from the tissue to the lungs. The CO2 is carried out in the front of HCO3 negative ion. CO2 plus H2O, this will form H2 plus HCO3 negative. Careful analysis of the same suggested that the partial pressure of O2 is high in the lungs, about 100 torr. This is responsible for the formation of HbO2. However, the low partial pressure of O2 in the tissues is responsible for the dissociation of HbO2. The dissociation of oxygen O2 diffuses into the tissues and oxidizes the metabolic fuel to CO2 and H2O. The carbon dioxide evolves in exhaled in lungs. Oxidation and oxygenation of Hb and Mb. The heme group consists of Fe2 positive ion embedded in a porphyrin ligand. The Fe2 positive ion has 6 coordination sites. The porphyrin ligands take up only 4 of the 6 sites, leaving 2 free binding sites on opposite side of the metal ion. If a free heme is present in aqueous solution, the 2 vacant sites may be occupied by the water molecule. In an aqueous solution containing oxygen, a free heme group binds to oxygen molecule in an irreversible manner with the oxidation of Fe2 positive to Fe3 positive and the oxidation of iron from Fe2 positive to Fe3 positive and destroy the oxygen binding activity of iron. The unproductive oxidation in a biological system is potentially fatal. The irreversible oxidation of free heme in the presence of oxygen involves a series of dimerization containing 2 Fe3 positive ions as shown in the figure. Thus, dimer is known as hematine. This dimer can no longer act as oxygen carrier. Deoxymyoglobin is a 5-coordinated high-spin Fe complex with 4 of the coordination sites of iron being occupied by the 4th nitrogen of the porphyrin ring, while the 5th coordination site is occupied by the nitrogen atom of the imidazole ligand of the histidine residue which join the heme with the protein. In the absence of O2, the ligand field is weak and such 5-coordinate heme complex of Fe3 are always high spin. The diameter of a high spin ion is larger than that of the central hole of the porphyrin ring. The estimated Fe bond distance is about 218 pm in high-spin Fe2 complex, while the size of the hole of the porphyrin ring is about 200 pm. As a result, the iron 2 Fe2 positive is compelled to 0.40 angstrom about the plane of the ring formed by the fourth nitrogen atom to give a square pyramidal arrangement, thus distorting the bond around the iron as shown in the figure. Thank you.